Thank you very much, Jen. Hi, everyone. Firstly, I would like to thank Jen and Wolf for having me to share my research to the audience of the ISNPR Knowledge Hub webinar. My background is a psychiatrist, working with patients with major depression for more than 25 years. I also lead the Mind-Body Interface Laboratory, focusing on clinical and preclinical research of depression for more than 15 years with my colleagues Jen. And being a physician and neuroscientist, I deeply understand the huge gap from basic research to clinical application, and we really need more translational scientists who work with patients to transform the research into patients' care. I think that's why we founded the ISNPR in 2013, and now it's a great pleasure to see the huge success that the importance of nutritional medicine has been well recognized as mainstream in psychiatry. Now, let me start my talk about the interplay between depression, inflammation, and omega-3 fatty acid. I'm going to start with the evolutionary perspectives linking inflammation and nutrition, and particularly omega-3 fatty acid to depression, and then the preclinical and clinical evidence to support using omega-3 for inflammation-related depression, and finish with future research direction. There is an evolutionary theory about our immune system with pro-inflammatory bias to explain the dramatic increases of depression and chronic medical illnesses in modern world, proposed by Miller and Reason from Emory University, published in Nature Review Neuroscience in 2016. In the left part of this figure, in hunter-gatherer era, early evolutionary pleasures of threats from pathogens, predators, and enemies resulted in the genetic selection bias toward immunological responses that conserve energy for fighting infection and healing wounds, as well as the genetic selection bias toward maintaining the alert and avoidance behavior responses, such as fear conditioning against dangers. In the middle right, as you can see, in ancient traditional environments, we exposed to minimally pathogenic organisms, so our immune system with pro-inflammatory bias can be held in check by the previously induced regulatory T and B cells, the M2 macrophages, as well as the production of the anti-inflammatory cytokines such as TGF-beta and interleukin-10. But in modern times on the right, prominent psychological challenges replace infectious challenges to activate our old defense systems without traditional immunological checks and balance in a more sanitized environment. Consequently, there is a significant increased rate of various inflammation-related disorders including obesity, dyslipidemia, metabolic disorders, and depression. Inflammation is not only important in depression, but also in several chronic medical conditions, and many risk factors for chronic medical and mental disorders can indeed activate inflammation. So where is the inflammation from if there is no injury or infection? There are some hypotheses. Firstly, early life and acute stress could activate macrophage and immune cells via sympathetic nervous system, the SNS, and the hypothalamus pituitary adrenal axis, the HPA axis. And secondly, as I mentioned just now, that some genetic and evolutionary factors can make us to be more inflammatory. And then thirdly, poor diet, lifestyle, and some systemic diseases could also induce inflammatory responses. And then you can see in the middle, the chronic low-grade inflammation then interacts with mind and body and increases the risks of diverse chronic mental and medical disorders. These conditions then could activate inflammation and further increase the risks of other disorders. In fact, depression is highly comorbid with several chronic medical diseases, and they share common biological mechanisms, including inflammation. Brain and body diseases interact as causal effect to each other. For example, the prevalence rate of major depressive disorder is about 5% in general population, but the prevalence rates are about 2 to 3 times higher in several medical illnesses like cardiovascular disease, cerebral vascular diseases, metabolic disorders, and cancers. And on the other hand, several well-conducted epidemiological studies also show that baseline depression was significantly associated with increased risks of above-mentioned chronic medical diseases. The information theory of depression is now one of the important and promising theories for depression. For example, clinical depressed patients manifest higher levels of inflammatory cytokines, and second, anti-inflammatory effects seems to be the common mechanism of all kinds of antidepressant treatments. And third, in the experimental model of human and animal, the pro-inflammatory cytokine can induce sickness behaviors and depression, including the most powerful supportive human model of interferon alpha-induced depression. And how did stress-immune interactions actually lead to depression? As you can see in the left figure, infection and injury leads to inflammation and cytokine production, and then affect brain function through 3 pathways, including first, the leaky regions of the BBB, the activation of autonomic nervous systems, and the direct microglia activation via specific hormone and the neural routes. As for nutrition, my lab is mainly interested in omega-3 fatty acid, which is probably the most studied nutrients in brain health and brain evolution. For example, in the left, an interesting paper in 2008 in Nature Review Neuroscience proposed that the excess of short-based diet high in omega-3 fatty acid had a key role to increase the brain-body mass ratio, also known as encephalization, which makes critical differences in survival and reproductive success when chimpanzees evolved into humans about 100 to 20,000 years ago. And then in modernization, the interplay between omega-3 diet and the brain is still ongoing. For over the past hundreds of years, our diet has dramatically shifted from high ratio of anti-inflammatory omega-3 to high ratio of pro-inflammatory omega-6 and saturated fatty acids. That is used to explain dramatic increases of incidence in many chronic medical and mental illness, including obesity, cardiovascular disease, metabolic disorders, and depression. The fact that omega-3 is important in depression comes from several lines of evidence. For example, in depression, a famous epidemiological study by Professor Hebron in 1998 in Lancet showed that countries with high fish consumptions to have a lower prevalence rate of depression, implying the protective effects of omega-3. In case control study, the lower omega-3 level has been found in depressed patients. In the meta-analysis we published in Biological Psychiatry in 2010 showed that major depressive disorder patients had lower omega-3 levels of both EPA and DHA. And if we looked at the non-clinical depressed subject without clinical diagnosis, meaning more heterogeneous group, the differences seems to disappear. However, epidemiological surveys and case control studies don't provide cause-effect relationship. The direct evidence from randomized control trial should be the gold standard. So in 2003, we published one of the first RCT to support omega-3's antidepressant effects. And in 2007, we published the first meta-analysis in Journal of Clinical Psychiatry to further support the clinical application of omega-3 fatty acid in depression. In 2012, we updated the meta-analysis in molecular psychiatry, and we found that the antidepressant effects is mainly from EPA, but not DHA. And for your information that EPA and DHA are two major components of omega-3 fatty acid, with EPA being more anti-inflammatory and DHA being more neuroprotective. And to our surprise, the antidepressant effects from EPA is consistently reported by several independent groups. Therefore, we started to think about personalized treatment of using omega-3, especially EPA, in inflammation-related depression. Here is a very interesting example. In 2016, in molecular psychiatry, Mishran and Rappaport stratified major depressive disorder patients with inflammatory markers, and they successfully increased the treatment effect size by using EPA in depressed patients with high inflammation status. The researchers' hypothesis here is based on omega-3's reverse effect of inflammation. Their finding is exciting because they prove that specific biomarkers may potentially push omega-3 fatty acids as the first-line treatment in certain patient groups. In fact, the idea of using omega-3 in inflammation-related depression has been supported in a series of our previous studies about interferon alpha-induced depression. Since 2006, we have been working with patients receiving interferon alpha therapy when it was still the only standard treatment for chronic hepatitis C viral infection. As the incidence of interferon alpha-induced depression is very high, some clinicians consider drug prevention. However, it's not clinically applicable to expose patients, especially most of the patients who don't develop depression, to unnecessary medications. In 2010, we published the lower omega-3 blood levels and specific phospholipase A2 and COX-2 genotype to be a risk for interferon alpha-induced depression. We saw that omega-3 is well tolerated as compared to drug. Therefore, we conducted the first placebo-controlled double-blind clinical trial to test the prevention effect of omega-3 EPA in interferon alpha-induced depression. There you can see in the right that EPA is the most effective to reduce the risk, while both EPA and DHA delayed the onset time of depression. Our study not only provides the important clinical possibility to change the treatment and prevention, but also confirms the notion that anti-inflammatory strategies are effective antidepressants in the context of depression associated with inflammation. We also want to identify inflammation subgroup not only by biomarkers but also by clinical phenotypes. So we investigated the depressed patients in two groups, including major depressive disorder and interferon alpha-induced depression, presenting physical comorbidity and somatic symptoms. And we found that lower omega-3 blood levels and higher inflammatory markers are associated with somatic symptoms in depression. This series of research linking omega-3, inflammation, and somatic symptoms are beautifully done by my colleagues Dr. Zhang. We also investigated patients with cardiovascular diseases with comorbidity of depression as a subtype, and we found that cardiovascular disease patients with depression had lower levels of omega-3 and omega-3 to omega-6 ratio as compared to non-depressed patients. We are also interested in how EPA and DHA may act differently in brains and in bodies. So we measure gene expressions and brain activation in depressed patients receiving EPA versus DHA. So in this study, we apply genomic studies. We compare gene expression on messenger RNA labels in 27 major depressive disorder before and after 12-week EPA and DHA treatment in randomized control trial. We found that both EPA and DHA decreased COX-2 gene expression, but only EPA increased the cyclosylic phospholipase A2 enzyme. In this study, we try to investigate why EPA is more effective than DHA in brain imaging studies. So we compare brain response to emotional stimuli in 24 major depressive disorder patients before and after 12-week EPA and DHA treatment, and we found that EPA, but not DHA, increased dorsolateral prefrontal cortex and ventrolateral prefrontal cortex activity when processing positive emotion. In addition, the increased activity in ventrolateral dorsal prefrontal cortex was significantly associated with the improvement of symptoms, and the increased activity in dorsolateral prefrontal cortex also revealed similar trend. To summarize, it seems that EPA and DHA have different actions in peripheral tissue of gene expressions as well as in brain functional activities. This is an interesting study collaborated with the main investigator, Professor Su Huanxin in Macau, showing the protective effect of endogenous and exogenous omega-3 on microinfections. By using two photon microscopes, the single arterial occlusion can be induced by the laser damage, and the rescue effect by omega-3 can be beautifully visualized in the left lower panel, and the molecular involvement of RIPK1 and the behavioral changes can be demonstrated. With the result of this study, we are now conducting a randomized control trial in depressed patients with microinfarction to see the therapeutic effect of omega-3 fatty acids. To summarize our work and others' work, I propose two applications about how to personalize the use of omega-3 in depression. Firstly, the biological specificity. We demonstrate that omega-3 is effective in inflammation-related depression, such as interferon alpha-induced depression and cardiovascular diseases. We are now conducting trials in chronic pains and obesity. We also applied preclinical models to investigate molecular mechanisms. Another application is safety consideration. We apply omega-3 in children, adolescents, pregnant women who are more sensitive to drugs side effects, or to prevent high-risk population to avoid unnecessary exposure to medication. To help the physician to use omega-3 in clinical practice, I invited the top highly cited researchers to work together in a panel of International Society of Nutritional Psychiatry Research to publish a practice guideline of omega-3 fatty acid in depression treatment. We conducted a Delphi survey and comprehensive literature review for the guideline, which is published in the Psychotherapy and Psychosomatics in 2019. This article is chosen as editor's choice with free access and provides the comprehensive information for treating depression with omega-3 fatty acid. This slide is to explain how I invited the authors. I used the citation index showing the top 10 cited principal investigators in this field of omega-3 fatty acid in depression, with me on the left, followed by Joe Hebrin, Robert McNamara, Kei Hamazaki, Maureen Freeman, David Michelin, Michael Mills, and so on. I was very happy that all the co-authors showed strong motivation to my invitation because they all agreed the clinical guideline should be very important. Here I just want to present briefly how the guideline looks like. We emphasized the practice should be based on comprehensive evaluation in clinical setting, and the quality of product is very important. And we agreed on using omega-3 in specific population, including major depressive disorder during perinatal periods, and children and adolescents, and also the elderly, and the prevention in high-risk population. In this guideline, we recommended the formulation and dosage, especially EPA for major antidepressant component, and recommend pure EPA or EPA combination with the ratio of EPA to DHA more than 2. We emphasize monitoring the adverse effect. In fact, the more carefully you monitor, the more adverse effect you will find. Although they are not major, they are only minor. I also want to talk about how clinical practice inspires preclinical research. Though we are now more confident that EPA might be more effective than DHA, but I think EPA might not be the answer, but hopefully leads us to the answer. Why? Firstly, EPA is almost undetectable in the brain. So I think EPA-derived molecules may be more directly to the therapeutic effects. My collaborator, Professor Richard Bazinet from Toronto, published many fantastic work about omega-3 derived mediators. So we started to work together from both clinical and basic sides. This is one of our collaboration to investigate a new target working with Richard. We hypothesized that omega-3 derived endocannabinoid, the ECB, might be important in the treatment of depression. So we randomized 92 eligible major depressive disorder outpatients in a 12-week double-blind clinical trial to compare the clinical outcomes on the left panel and the biological markers on the right, before and after EPA, DHA, and EPA-DHA combination intervention. We found that EPA-derived endocannabinoid, the EPEA, seems to be the key marker for EPA's antidepressant effects, meaning EPA works when EPEA levels go up. This is another example to show omega-3 PUFA-derived lipid mediators may be also important. Dr. Borsini used hippocampal progenitor cell lines and found both EPA and DHA prevented the apoptosis and neurogenesis reduction. In her sophisticated experiments, she showed that the effects were mediated by the EPA and DHA lipid mediators metabolized by the lipoxygenase, the LOX, and the cytochrome P450, which we further used major depressive disorder patients in DHA and EPA clinical trials to validate the result. My colleague, Dr. Jian Zhang, and I recently guest edited a special issue for the BBI Brain Behavior Immunity, Nutrition and Immunology in Mental Health, Precision Medicine and Integrative Approaches to Address Unmet Needs in Psychiatric Treatments. We included 15 papers discussing the role of not only omega-3, but also blueberries, melatonergic agents, cannabinoid, in the context of immunoregulation of brain behavior and immunity. From omega-3 derived mediators to many other nature and nutritional compounds, I think future research might find many promising targets to develop novel antidepressant agents. The COVID-19 pandemic changes the world. It's not just the viral invasion to the lung or specific organs or a systemic infection. It's also a whole cycle neuroimmunity model to the brain behavior and immunity. At the beginning of the outbreak in March of 2020, I initiated a special issue about COVID-19 in the Journal of Brain Behavior Immunity. We collected 93 papers in 4 months, which collected huge citation on this issue. The pandemic involved both body and mind. The direct neural invasion of COVID-19 induced olfactory and gustatory impairments and severe encephalopathy. In addition, the COVID-19 triggers the cytokine storm that becomes the main fatal cause of COVID-19 or less severe consequence of systemic diseases and inflammation that increases risk factors of mental disorders. And psychologically, during the COVID-19 pandemic, the general population, the infected patients and their families, and even the medical staff all feel anxious and depressed under the stress of pandemics. Furthermore, the lockdown has tremendous social and psychological impacts. So we published this review article to demonstrate the interaction among brain behavior and immunity of COVID-19. Regarding to prevention, vaccination may be the best strategy, but it doesn't provide full protection. We could not overemphasize the importance of healthy diets, healthy lifestyles, exercise, and nutrition. We recently did an extensive literature review and published the article about the role of omega-3 fatty acids in the psychological and physiological resilience against COVID-19. Recently, we know that COVID-19 infection could lead to the prolonged sequela of heterogeneous negative impact in multiple systems known as the long COVID. Literally, the scale of long COVID is global and extensive, and I concluded in this new article in BBI that omega-3 might have a role in the prevention and intervention for long COVID. Finally, let me introduce to you our annual conference, the 12th Mind-Body Interface International Symposium. This meeting has been about translational medicine, covering nutrition, exercise, yoga, meditation, and even music therapy. So please join us. I'd like to stop here to acknowledge the founding bodies, my collaborators, and most importantly, my laboratory colleagues. And thank you very much for your attention.